Okay, welcome everyone. I hope you all had a great break and are now happy back in this and have started well in this new year. So welcome to this first joint Broad MIT EECS Colloquium of the year, which is hosted by the Eric and Wendy Schmidt Center in AI+D within EECS at MIT. So I'm very excited that we have today Gunnar Ra here, who will really tell us about going this two-way street of working on important biological questions, and then also have important biological questions motivate new foundations of machine learning. So Gunnar is a professor in the computer science department at ETH Zurich, where he leads the biomedical informatics group. Gunnar got his PhD in the German National Laboratory for Information Technology, and then he was for some years a postdoc and also an independent investigator on working on machine learning and genome biology at the Friedrich Miescher Laboratory in Tübingen. He then moved to New York, to the Memorial Sloan Kettering Cancer Center, before he then joined ETH Zurich in 2016. So his research really spans all scales of biology, so all the way from proteins to cells to tissues and to organisms, and that then motivates different foundational developments in machine learning. I think today we'll hear a lot about distribution shifts and the importance of it, of course, when we're thinking about medical health records, but also, of course, for the others who work more on cells or tissues, we also have a lot of different problems around distribution shifts. So I'm very excited for Gunnar's talk, so the floor is all yours.
Right. Great. Thank you so much for the very nice introduction. It's a great pleasure to be here. I had very intense and interesting discussions already, so it's awesome. And now I'm really looking forward to tell you a bit about the research which we do. So let's get started. I thought I'd start with a slide. This slide is actually 10 years old. It kind of motivates a lot of the work which we still do, and I think there's still a lot of truth in this slide. So it was part of a BD2K cancer AI grant, which in the end wasn't funded, but it doesn't matter. And it still has the main components. We need data representation, depending on the data modalities. We need something like a joint model, which combines all that data and makes predictions. And then we need things which are sitting on top of that, which allow us to make some actions, right? So to have something actionable based on that. So a foundation model alone is not sufficient if you can't do any actions on top of them. So these are the three different things which we think about. So how to deal with electronic health records, how to deal with pathology images, with genomic data, with drug information, and so on, and how to bring this all together. So, and it motivates a lot of our research. You will see two cases which I talk about where we kind of instantiate some bits of that, right? And yeah, it sounded all pretty easy in 2013. We want to do that, but it's taken 10 years now already, and it's still ongoing. Okay, so currently we think a lot about foundation models. And we think about data which comes from different sources, from EHR, from pathology, from genetics, and so on. And typically what we do is we distill information from each of these data sources into some interesting bits which we find interesting relative to let's say, human disease. So and then, let's say we take these bits and put them on a time series, and we would like to predict, or maybe would like to absorb information until a certain time point and get to a patient health representation, something which summarizes all that which we have seen, and so we can actually make predictions based on that. So this is a very useful tool, right? So it's a universal tool to summarize patient data. It's compositional because we can add maybe new data modalities to it. It can be trained unsupervised if you have this time series data long enough. And it is hopefully predictive of future health states. And so these are these multimodal foundation models. So this is, I think, some of the ideas which we had early on. And I still think these are really important to implement in the end. But we have to see whether they work in reality too, right? So, and I show you some cases where much simpler methods work much better, and that it needs maybe much more developments on these methods to make them really work in real cases. Okay, good. So I will talk about two things. One is not molecular. It is in the intensive care, early warnings, early alarm system for organ failure in an intensive care unit. So time series data, time series models, of course, and then we have a lot of distribution shifts when you go from one hospital to the next. And then I spent a bit more time on multimodal profiling to advance cancer treatments. This is the Tumor Profiler Initiative, where we have bulk, single-cell, spatial, perturbation data, and multimodal models, and also quite a lot of distribution shifts. Okay, so let's get started. So intensive care. So here the data is, we have electronic health records, we have drugs, and we have ICU monitors. This comes in over time. And what we would like to do is we would like to have models which allow us to make predictions in the end about organ deterioration. So does, for instance, the circulatory system deteriorate or the respiratory system deteriorate over time? Or maybe we could also use it in order to predict which drugs should be used in order to maximize survival. So we haven't done that, but it would fall into this category. So the question is, how can we best model health states and treatment effects? How can we make AI models useful and ready for the ICU? And can AI-based systems eventually benefit patients or the healthcare professionals? So these are important questions. So the setting which we look at is the intensive care unit. This is a case where we have actually a lot of data which comes in all the time. So these are the monitors and drug dispensers, and all this information comes in all at the same time. And what happens is that each of these devices can give off an alarm, and there's an information overload of physicians and nurses to deal with all this information in order to actually make that prediction, is something happening with this patient or not. So there's information about the circulatory system, the pulmonary system, the neurological system, the renal system, and infections. And we get in essentially intense data streams on that. So let's look at one case here. So, and this is from an earlier publication, and this is on circulatory failure. And it's defined as a sustained condition of a sustained period of low blood pressure with elevated lactate. So low blood pressure, elevated lactate, right? So, and this is the case when it actually happens. So this is easy to check, right? So you measure lactate, you see blood pressure low, and then you can say, okay, that's the circulatory failure. That's not what we want to predict to be in that time, but we would like to predict it in advance, right? So that's important. So, and the setting which we discussed with the physicians is that we would like to be able to predict on a currently stable patient, so somebody who doesn't have circulatory failure at the moment, whether he or she will get worse in the next 8 hours. So worse meaning any time in the future in the 8 hours. The 8 hours is chosen, so it's shift length, and we make predictions every 5 minutes, so quite often, right? So that we don't miss it. So the idea is that essentially the data would flow into the AI system in real time, make these predictions, alarm the physician. The physician then may do some adjustments to the treatment of the patient. So it's not, we don't decide anything on the treatment, right? We just put the attention to the physician. And that's important. I mean, if the physician had time the whole time and sit next to the bed, they would do a pretty good job. But typically, they don't have that, right? So they would do something else, and I'm not saying forgetting, but don't put full attention to that patient. So such a system can be really useful. So coming to this. So let's say as computer scientists, we sit here, and the clinical practice is usually quite different from what we think about. So, and there's a high demand of interdisciplinary collaboration and cooperation and understanding. So what I explained to you before is not something the physician told us exactly. It was the result of a discussion among us, right? So what can we do? What can we model? What is interesting? What is relevant in the ICU to be actionable? So I think we really need to communicate very closely to understand the problem on the other end. that we develop something which matters. So there's a lot of machine learning in the clinical side out there, probably hundreds of papers, and only 1 or 2% of them ever make it on the other side. So because that communication isn't quite happening. Good. So let's move on. So now we have done this. We have received essentially data from the intensive care unit in Bern. We got essentially a whole dump of their database. We had to deal quite a bit with the data quality and so on. We did all that. And after that, it's quite standard machine learning. So we did some imputation. We annotated the state for whether there is circulatory failure or not, right? And then next step, we do supervised learning. We extract some features. We do the labeling based on that, and then we do supervised learning and do some evaluation. So I would say the machine learning here isn't super technically difficult. It was a lot of work to get the data and getting the labels right and all that, right? But so technically, this wasn't super complicated. Nevertheless, I think what came out was pretty good. I think one novelty or one complication is how do we evaluate these kind of predictions? So think about, and this actually took a lot of time to get right. So this is an event. So let's say where circulatory failure happens. So this is our score. It goes up and down over time, right? And let's say this is a threshold which you have chosen. And if the score is larger than threshold, then we raise an alarm, okay? So this here would be a false alarm because it's too far away from the event. This would be a true alarm because it is close to the event, it's within the 8 hours, right? So this is a captured event because there is an alarm before. This is a missed event because there was no alarm before, right? So this way we can compute the precision and the recall. So the precision is for the alarms and the recalls for the events. Ideally, we catch all events and we have as few as possible false alarms, right? So we can compute a precision recall curve, and ideally we are interested in points like 80 or 90%. We would like to get maybe 80 or 90% of the events predicted correctly at a high precision. It doesn't look very high, right? So I mean, precision 40, 50%, but still it's clinically pretty useful. So that means every second alarm is probably correct. But there's some good reasoning why this may be low, because it could well be that you do an alarm here, the physician does something even without the alarm, prevent the actual circulatory failure, and the alarm wasn't wrong, it just was prevented, right? So there's a few reasons why the precisions could be low here. Okay, so when you compare that with the number of alarms which typically would sound in the ICU, this is whenever the physiological value is out of range. So you get, on average, I think 10 alarms per hour. With our system, you get, depending on whether you have circulatory failure or not, a 0.1 to 0.5 alarms per hour. So over the whole day, maybe 1 alarm per patient, roughly. So, I mean, it's much lower, and therefore the saving, I think, in effort in looking at after the patient is much lower. So here's one detail. So we trained this on Bern data. So this is a hospital in Bern in Switzerland. And there's also data from Boston, from Beth Israel, the MIMIC data set. And we validated the score on that data set. So essentially, we just tested, is the predictor predictive in this setting? And it turned out the performance was lower. Okay, maybe not totally surprising, but it was a bit lower. And it turned out, after a little bit of digging, that it is related to the resolution of the time series. The MIMIC dataset only has a 1 hour resolution. Our dataset has 5 minutes resolution or even 2 minutes resolution. If you downsample our dataset, you get exactly the same performance. So this was really nice because we identified the source of that problem. And in the end, the predictor just works equally well in Boston as in Bern. So nice. So that's not always the case, by the way. Good. So we have developed this validated machine learning system, validated in another hospital. We have also done this now for respiratory and for renal failure. The performance depends a lot on the time resolution we have shown and also the data set size. So if you have more data, it becomes a bit better. The variables and interpretations are all plausible. The system performs well enough to be of clinical utility. So this is all nice. But now the question is, does it make any difference for patients? So that's, I think, one question you should ask. Maybe you ask, well, I've been asked, isn't there any deep learning here, right? So, and there isn't, but I tell you a bit why. So let's get to the first question. So, so we design a study, a prospective study, where we have a silent running of the system, where essentially we first measure whether the system prospectively works equally well as it was prospectively. So we could just do this without any interventions, right? So the alarms are not given to the physician and there's no harm. So it's a relatively easy, technically still, well, a lot of effort, but it could be done. So for 2 months, we let the system run and then see whether the performance is the same. So hopefully it will be. So implementation is ongoing for this. So the next one is, the question is, how do these risk scores compare against risk scores from physicians, maybe the treating physician? So you would like to ask the physician, give us your risk assessment. And then we can compare what's the risk assessment of the machine versus the risk assessment of the physician. Or maybe even, how is the risk assessment of the physician if we give them the score in addition? So maybe they actually get better, right? So maybe this is a non-technical study, but very interesting. And then the final bit is we do need a randomized control trial to really say this alarm system does make a difference. So we need to randomize and say maybe half of the patients do get the alarm system, the other half doesn't get the alarm system. And we did some calculations. We need about 1,000 patients for this, and then we can actually do this study. It's not powered enough for mortality, but for the number of events, reducing the number of events and so on. Good. So we have developed an app. It runs on the physician's phone. It gets a real-time data stream from Epic and then computes the risk scores in the back end every 2 minutes. And then we get an alarm push notification to the phone. Good. So now through the second question. This is about deep learning and maybe performance of algorithms. So first thing which we need to do is we put the data out as a data set which people can download and use. It's the HIRIT benchmark data set you can download from Physionet. It's not only that we put the data there, we put also our data definitions, our processing, everything. So, so it's completely reproducible together with our labeling. So people who work on machine learning can try new methods on that. Then we also ran a set of algorithms on that. We defined tasks for circulatory failure, respiratory failure, kidney failure, and so on. And then we ran different standard algorithms for these tasks. So logistic regression, light GBM, and then some deep learning methods, including transformers and so on. And it turns out they don't work as well as the simple method, the light GBM method. So this is what we have used in the original paper, and it just outperforms all the other methods on all tasks. So, I mean, this is structured data, right? And deep learning methods have more issues with that. But still, we would like to advance machine learning, right? And we don't want to advance light GBM, or, well, we are interested maybe more in those methods. So this is motivated to do a lot of new technology or new techniques to make these methods better in the end. So, so we worked on different aspects. We worked on better models of temporal dynamics, dealing with the missingness, dealing with the heterogeneity, with different time scales, the interpretability bits, domain shifts, and so on. So there's each a bunch of papers related to that. So many of these advancements are based on deep and Bayesian learning. I cannot talk about all of them, and I shouldn't, but just want to mention 2 different things. One is the domain shift between hospitals, and the other one is the time to event predictions, which we look at. Okay, so this is a different task. This is respiratory failure. This is the performance on HIRIT, Bern. This is the performance on Amsterdam data, okay? It looks hugely different, huge performance difference. The question is why, right? So, I mean, we didn't know why. So, and we looked at it in many different ways. We tried to normalize the variables and try to account for some different ranges and so on. Didn't help. What helped is understanding which variables are responsible for it. And let me explain you this plot. And this took a while until we figured it out. So on the x-axis are the variables which are used in the prediction. And they are sorted by how much performance they increased on the source data set. Okay, so on the original data set. This is this performance. And you see the performance is going up, right? So because we do forward selection of these variables, so the performance should go up. So in green, you have the performance on the other domain, meaning the other test domain. So that's the Amsterdam data set. And you see for many of the variables, it goes up, but then there's a subset of the variables where it goes down. Right? And this is where the performance is affected by including that additional variable. So that variable must be one of the reasons, at least, why performance goes down. And if you look at them, these are all treatment variables, almost all. I mean, there's also gender in there, but most of them are treatment variables. And the physiological variables or the ventilator variables, they are not in there, typically. So, and if you do that, then the performance gets much closer. I mean, it's not fully recovered, but much closer. So it appears that the generalization really depends on the origin of that variable. Where is it measured? How is it affected? And so on. And if this is something which is physiologically present, this is probably the same in Amsterdam or in Bern, right? Then this is fine. We can include that. But if it is maybe dependent on treatment, may have different policies and so on, then we better don't include that. At least in respiratory failure, there was a strong effect on that. Circulatory failure, it wasn't. Okay, so maybe if you have relatively low-dimensional data, so then you can do this kind of plot as a diagnostic plot, you could say, and you find out which variables are the problem. So, and this helped us in many cases. Okay, so just to mention a second approach here. So how's the time? Okay, second approach. This is a temporal label smoothing. This is about you know, when you have the event here, then at different, let's say this happens here, at different time points before that event, this is all a true label. And when you go from 8 hours to 8 hours, 5 minutes, it's a negative label. So here it's a false positive, and here it's a true positive, right? It feels a little arbitrary. The 8 hours is not super well chosen, right? So the idea is simply that the further you go away from the certain time point here, the lower the weight for that label should be, right? So, and you essentially have some decay curve which depends on the distance to the event. And this smoothes out the label. And so then it is not too much confused here. So that's the idea, right? So, and that leads to not huge improvements, but it is consistent improvements in many cases, which we have looked at. So usually about 1% in performance gain. Good. So these are just two examples. So what we would like to do really is to develop these foundation models. As I've described, there's many challenges with heterogeneous structured data, incompleteness of the information, and so on. But I think we need to advance methodology to really bring this forward. So the question is, these ICU foundation models eventually, at least, outcompete the standard, the classic machine learning techniques in the supervised setting. And we work on that. So we have now an ongoing project where we would like to train, let's call it a foundation model. It's on 500,000 ICU patients, which is a combination of different data sets from different hospitals, and combine that with prior knowledge, add a bit more modalities, for instance, text, and then try different architectures, different techniques to fine-tune these models and so on. So we need quite a bit of compute for that. Happily, we got access to the Swiss supercomputing system, which gives us a lot of GPU hours to do that. So thank you. Good. Now let's switch to the second part. So yeah, it's time. Good. Tumor profiler, part 2. So this is completely different. Forget everything I said. It's on cancer. And this is a multimodal tumor profiler study. And it is a major project in the sense that it received a lot of funding. It is just 240 patients, not 240,000, that would be great. And we have actually a lot of different techniques for profiling these patient samples. So we have electronic health records, digital pathology, targeted NGS, cell-free DNA, single-cell genomics and transcriptomics, bulk RNA, proteomics, CyTOF, imaging CyTOF, pharmacoscopy, and this is a deep drug, what we call it, is also an in vitro screen for testing. Good. So the setup of this project was interesting, I think. So the idea was that we get this data within 2 weeks for each patient. So we have 240 patients. We have essentially 240 iterations through this pipeline. And for each of these modalities, there was the group who generated this data wrote a little, let's say, research report about this specific patient. What was specific about this cancer with respect to that modality? Okay, so maybe EGFR high or something, whatever they could measure in their technique, right? Or maybe a high immune cell infiltration. So this went into a molecular research report. Then this was discussed jointly with an oncologist who knew a lot about molecular biology in a pre-tumor board, and a molecular summary report is produced. This then goes into the tumor board and decisions are recorded. So ethics didn't allow us to really do all the treatment decisions if they were different from standard of care, but for a subset of the patients we were allowed to do that. So, and then we recorded for each of these patients what the treatment recommendation would have been if they had only standard of care information. There would be, let's say, pathology, or maybe a bit more than standard of care, in Switzerland at least, pathology plus NGS, or if they had all the other information in addition, right? So, and the question is, how much of the decision-making was influenced by this additional information, right? And maybe which markers or which technologies actually contributed to this, right? So this was an interesting exercise in understanding that. So this happened now all within 4 weeks, so it could really influence the decision for some patients. And now what we are doing is also a full cohort analysis and do some new biology on that. So so we started off for each patient with about 500 gigabytes of data. There was a lot of imaging data, and there was a lot of automatic processing, which also involved some machine learning and so on. But in the end, it produced this molecular research report, which was a mix of algorithms plus humans looking at it, at plots and so on, right? So then there was really mostly manual decisions after that in the discussions, whether this is relevant or not relevant. So, but the nice thing about this, we recorded the different stages. We knew what information was used by the oncologist at the end to make that decision. So for each of these patients, we have essentially a ranked list of drugs, together with supporting information, which of these technologies or modalities supported that specific drug choice or not. So, and we had 120 melanoma patients, 80 ovarian carcinomas, and 40 AML patients. So, I mean, they're all not huge numbers, right? But since these were 12 different technologies, it was not scalable to larger numbers, let's say. So let's look at the melanoma cases. So here you have about 120 patients. We looked here into an analysis of the clinical utility of this decision-making or of this additional information. And there were the adjuvant patients, the palliative standard of care patients, and the beyond standard of care patients. And the beyond standard of care, there we have some, let's say, some flexibility of how to treat them, because it's not defined very well in the standard of care. So, so we have about 36 samples here, or patients. So now, so we do this decision, and then we compare how often did the tumor board in the end decide for the treatment recommendation, which was based on pathology only, pathology plus NGS versus all information. So at least what we observe is that the tumor board more often decided for the treatment recommendation, which was given based on all information. It doesn't mean it's a better one, right? But at least the tumor board was convinced that there is relevant information in there. So we also did some analysis of a matched cohort where we took the tumor profiler cohort and matched it against a set of patients which had the same characteristics. And then we could do some survival differences. And it is significant, barely, but it's, or maybe it's barely not, but roughly significant. But in the end, it needs a randomized control trial. So, and that's what's starting now for ovarian carcinoma, where half of the patients, or randomized, half of the patients get the tumor profiler test, and the other half doesn't, for a subset of the information. Good. So the question is, which information was actually used, and can we maybe work with less, right? So I mean, from a diagnostics point of view, so it's pretty expensive to do all of this, but maybe we can do less. So what you see here is a recording of the different types of features which were used by the physician in order to make the decision, okay? So, I mean, the PERC, for instance, was used a lot and can be provided by different technologies. And also PDL1 can be analyzed by different technologies. And then there's some which are pretty rare here. And then what we did the following is we looked at all combinations of technologies, all subsets of technologies, and asked, could a subset of technologies give us the same information, right? So could we do the same decision on that, right? And each dot here is a combination of technologies. And I guess interesting is this curve here, right? So this wavefront here, which tells me that maybe among the 88 samples which we have, maybe 69 samples we could have done with 6 technologies. On the x-axis is actually the cost for the measuring, and the y-axis is the number of samples which would have the same decision. Okay, so ideally you would like to have something which is low cost, plus it doesn't change the treatment recommendation. Okay, so if you have all the technologies, maybe it's $10,000 or more, right? But maybe with the subset, you can do with $2,500 or something, and most of the decisions would be the same. So this is an interesting analysis, I think, into the cost analysis to understand really which technologies are useful.
Sorry, this is the same technologies per patient, or are you allowing yourself to switch which technology?
No, it's the same set of technologies. So that needs to be fixed in advance, yes. Mm-hmm. Yeah, so otherwise, of course, I could just choose the right ones if I knew in advance, yeah. So, good question. Good. So now, obviously, what you would like to do, I mean, you would like to be able to predict these rankings, ideally, right? So, I mean, it would be a nice, interesting machine learning problem. So could we maybe take all the data which we get from a patient and predict these rankings? And maybe can we mimic the oncologist? So I think the first part, that's what we can do. We can distill maybe the relevant information in multiple stages, maybe reduce the sample analysis costs overall, maybe by being able to predict one modality from another modality or these kind of things. So that's feasible, relevant, machine learning, interesting in it, right? But can we learn to imitate the oncologist and maybe rank these treatments? I think it's still very hard. So, I mean, with these sample numbers, at least, if you had 100,000 samples, maybe that's doable. But with 240, I would say that's impossible, close to impossible. We have tried, so it doesn't work so well. So we focus on that, right? So we don't even try that because there's not enough data. Good. So now, as somebody who develops methods, this is a great data set. So you can take any combination of modalities here and develop a new method, you could say. So we have done this. For instance, we worked on a method SIM, which does some single-cell integration via deep learning and matching for single-cell transcriptomics and cytoph data. Here we have a method which predicts from digital pathology and IMC. It predicts protein expression from H&E, I mentioned briefly. Here, there's a method which integrates spatial RNA-seq and H&E for learning embeddings. Then here's a technique, CellOT, which takes the single-cell perturbation data here and combines it with other information like the electronic health records. And here we have a method which is path of A, which is probabilistic pathway multimodal factor analysis method. So, I mean, essentially, there's lots of possibilities here, and I cannot talk about all then. So I briefly mention an outcome here, a little bit of the technology here, and a bit more here. Okay, I think this is interesting to you. Okay, so how can we do this for? How can we combine H&E and IMC data? So H&E and IMC, they work both on FFPE slides. H&E is just the image, the stained image, and the IMC is protein markers which are measured with CyTOF. And we have about 40 proteins which could be measured, which were well selected for melanoma, for instance, but relatively small patches. So it's really a small part of the image. Yeah, and I mean, it's multiplex, so that's good. I mean, it's not RNA-seq high dimensional, but it's reasonably high dimensional. And the question is, can we predict maybe one from the other, right? So the idea would be that if you had a model which can predict from the H&E, that you get a whole slide image with multiplexed protein abundance. And that could be very interesting because maybe in the end, we don't need to have the IMC measurements to be clinically useful. So I don't talk about the method because I don't have enough time. I just want to show one example here, one use case. When you do this, you could apply this now and predict the markers, for instance, CD20 for B cells or CD8A marker for T cells. And you could see that in that region here, in the tumor region, these B cells and T cells are present. So it's a highly infiltrated tumor. So it's a hot tumor, which is very relevant for cancer treatments. So this one here is a cold tumor, and you would probably not do certain types of treatments. So it does work pretty well. It does have potentially clinical impact because we can just look at these low-cost images and predict where all the immune cells are going on and more. So that's interesting. There's also an interesting method in there, but I don't have time. So here's another approach. This is relatively new still and not published yet. This is a technique where we do learn embeddings between spatial RNA-seq data and H&E. So the idea would be that you take image features, so PCAs of image features, PCA of the RNA-seq features, reduce dimensionality, and then have a spot level presentation together with the neighborhood around it. And what you would like to do is you would like to learn an encoder which takes this information, which can decode and reconstruct the original. But in addition, there are some additional losses here. There's a triplet loss where clusters on the image features should be recoverable from the latent space and also on the spatial transcriptomics end. So in the end, these reconstructions were based on these latent space. You would like to do maybe clustering or some additional analysis, right? So we really care about the latent space and the geometry in that latent space, whether that measures the right similarities. So the method has two parameters. One is essentially a weight in front of this part, and the other one is the weight in front of this part. So how much weight do you put to the image features versus the expression features? And these may be task dependent, so they need to be tuned. So now, what we have done is we looked at a few data sets which came with existing annotations. So essentially regions which were clustered in certain ways. So this is tumor or so. I'll show you some example. So we just took these annotations and asked, is a clustering on our latent representations reproducing these annotations which already exist, right? So and this is what we measured against. So with the adjusted RAND index. And here are different methods which we compared against. So this is our method, and you see, well, it works really well. But important is that these parameters were tuned for each of the tasks separately. And that's what some of these methods don't do. And I think this is key. Some of these methods use all sorts of information. Some just use transcriptomics, some use spatial plus transcriptomics and so on. But in any case, it works really well on all of these data sets compared to all of these methods. Here's just one example for tumor profiler, metastatic melanomas. This is a segmentation which was done by pathologists. This gave us the annotation, right? So, and then the question is, can we now run our algorithm, get the latent representation, cluster these latent representations, and how well is it agreeing with this annotation, right? So, and this is what we get by our method. This is what we get by Muse, another algorithm to do that, and the concordance is much, much better. So this is nice, but I think it's really important that you have ways to tune these parameters alpha and gamma. Good. Now, let's get to the last topic. Yeah, 10, 15 minutes, good. So that's just enough. So this is using data, a single cell drug response data. And this is a collaboration with Charlotte. She probably was here and talked about it before. Stefan Stark in my lab, Gabriel Good, who's done a lot of the measurements, but also statistical analysis, Kyong, and postdoc in my lab, Lukas Pelkmans, the lab who generated the data, and Andreas Krause, who will come for sabbatical soon. So the question is, we do measure here the in vitro drug response for many cells. The question is, can we predict the single cell response in advance for new cells? Can we also do this for new patients, right? And is it predictive maybe of therapy success? So here's the technology to measure it. I don't want to go too much into detail. It's a platform which allows you to measure via imaging many different features, in particular marker expression and morphology features of the cell. So in the end, for the tumor profiler data set, we had 37 patients with 34 different drugs and 7 intensity markers and 15 morphology markers. Those intensity markers were chosen to be relevant for melanoma. Okay, you find PRKT and a few other markers which are really relevant. Okay, so this is the data. This is maybe a rough description of what kind of samples are in there. These are all melanoma patients, but they come from different sites. There are different biopsies. There are sometimes metastases, different subtypes. Number of treatments is very different. So it's really highly diverse set, okay? Which can be good or bad, but it depends. Maybe here's the speed to the progression speed of that tumor. So cell OT, maybe some of you have seen the paper or have heard the talk here. So very briefly, the idea was that we have a set of cells here before treatment, we have a set of cells after treatment, and we would like to learn that mapping, which goes from an untreated cell to a treated cell, this mapping T. And we would like to have a parameterized function where we put in essentially the description of a cell before treatment, and it gives us the cell after treatment. And essentially, we look for a mapping such that that distribution is identical to that distribution. Right? So, and this is then our treatment effect, so as you can see. So don't want to go too much into detail here. This is based on optimal transport. You minimize some variation, how much that mapping induces a change in that cell, and you would like to minimize that overall transport maps which do this shift. And you can parameterize this with neural networks, with input convex neural networks, and the gradient of that is the transport map. So let's take this as given, and let's look how it works and how we measure that it works. So we looked into 3 different data sets. One is the data set from Tumor Profiler. This is a cell line mix. This is the melanoma cohort, and this is a single cell RNA data. So there are other methods which can do this. So cell OT is one, a CGen, a CAE, or just moving the average. So what we measure is the MMD, the maximum mean discrepancy. This measures how different the distribution is before, predicted treatment and the observed treatment cells, right? Good. So, and here we have used SCGen embeddings. Good. So let's look at this data set here and just show you how well it actually works. So so this is a drug treatment with imatinib, and this should affect PAKT and pERK. So, and what you see here is the distribution before treatment, and this is the distribution after treatment. So when you just move the average, then this distribution would move here, and it wouldn't be a good match. If you take a method like it would, maybe even overshoot a bit. And if you take cell OT, then the distributions match really well afterwards. So it's not cherry-picked. It's in many cases in IID setting, it really works that well. In out-of-distribution setting, it's a bit harder. So let's think about what we can do with that. When you think about all treated cells with all these different drugs, and you plot them, you get a relatively mixed bag of drug responses. While treated cells, they're all over the place, right? So here the color is the drug, which is the treated drug, and you see all the treated cells. What you see on the right is the difference between the predicted drug treatment minus the original cell. So that's essentially how much change is there happening in the dimensions which we care about, right? So and if you do then a UMAP, then you see that the different distributions, they really, or the different treatments, they really separate really well. So this becomes less clear when we have patient effects. So this is all on cell lines. I'll show you that in a minute. But at least this is reassuring, right? So we can really see what the effect of the individual drug is. So it becomes more complicated, and this is now new data on the tumor profiler, and this new results on the tumor profiler data. So here's maybe the cohort variability, and we can see that it's really dominated by patient uniqueness. So what you see here is just the first two principal components of the average cell, okay, averaged over all cells for a patient. These are control cells, and all the other dots, these are treated cells. And you can see that, the control cells are all over the place, and they're mixing really well with all the treated cells, and it really spans the whole space. If you then look a bit at drug effects in this PC space, then you can see a drug like combination like trametinib plus dabrafenib, which is a strong cocktail of drugs. It's a strong effect. You see, there is good movement in the first, maybe in the second principal component here, and it's really maybe somewhat consistent, right? So if you go for a drug which has a less strong effect, then you see maybe differences in principal component number 4, but not so much in the first ones. And if you go for a weak drug effect, then essentially there's no movement in the first 4 components. So now let's look at combination. Essentially, it's a convolution of the drug effects plus the patient effects. So if you look now, these are all cells. It's roughly 8 million cells. These are all the observed treated cells. And it's just the colored ones are only the ones which are done with this strong drug combination. And it's colored by patient ID. And you can see it really covers the whole space. So this here is the single cell drug response. This difference between the treated or the predicted cell minus the control cell, the pair, right? So, and you can also see it's still quite distributed, but it's much better. It's much more localized. And you can measure that. You can look at the k-nearest neighbors and ask whether the k-nearest neighbors in that or that plot would have a similar label, namely the same drug label, right? And you can see that after you transform it this way, the treatment label gets much more similar to the neighbors. So here it's 50% of the neighbors have the same treatment label, whereas here it's only maybe 5% or 10%. So it makes a huge difference. So I think by looking at the single cell drug responses, we get some of the patient variability out, which is important, I think. So, so let's look at the drug responses and compare them among patients. So what you see here is, this is patients by patients, okay? And the color here is a function of the MMD. A large color means very similar. Low color means it's a large difference between these two patients. This is based on the treated cells, the distribution, and this is based on the single-cell drug responses, okay? So these differences. Okay, so I mean, quantitatively, maybe the maps, I don't know, maybe they don't look too different, but what is really striking is that if you look at clinical features like driver mutation status or other features, then you find much more clinical associations when you look at these plots than at these plots. So here you can see this subgroup here of patients behaves very different from all the others, and these are the patients which have a V600E BRAF mutation, right? So which is important for this drug cocktail. So you can formalize this and do this analysis over all drugs and all these features, and you essentially find that when you try to associate it with the observed treated cells features, then there's few associations which are significant. And if you do the same analysis with the single-cell drug responses, then there's a much more consistent and much more stronger signal, which we can see. So we have seen this, for instance, much stronger associations. And then we have seen this for this drug I've just shown you. Here's another drug, paclitaxel. And you can also look a bit what are the differences which are produced by this drug. So these are the control cells. You see essentially NRAS status. It's not, levels, features are not very different. But if you go into drug response, then the NRAS status really changes how the drug changes, what the effect of the drug is. So essentially, that drug changes tubulin formation. Okay, last result slide here. So can we now look at that treatment response, this single-cell drug response, and use that information in order to predict the maybe time to progression for patients, okay? So we classified it in two different categories, fast or acute, intermediate, and slow, okay? And then we would like to predict essentially in a multi-class setting based on the cells which we have, is it in one of these three classes? So first we look at IID setting. IID setting is when we train a cell OT on the same patient. So we essentially have seen the drug response data for that patient, right? And then train that map and do the calculation of the single cell drug response. And then we see, okay, if we look at this feature here, okay, maybe the legend is missing, okay. So this one is here is the single cell drug responses, which are computed by cell OT. Then we get a signal we can at least predict a bit better than random. So it's not a huge signal, but it is significant. And if you look at patient mean features, meaning taking averages over all cells or taking some of the other methods to compute these transport maps, then it doesn't work as well. So this same remains if we look at the OOD setting. So we look only at the control cells for these patients and use a transport map which was computed on other patients. So the performance still remains reasonably high. Okay, good. I think we are essentially out of time. Let me just make a few more points. So the multimodal tumor boards show a lot of promise. We can make a difference in the tumor boards in terms of decision making, but really whether it makes a difference for patients, we need a randomized control trial. So the large-scale single-cell multi-omics datasets bring many opportunities for methods development. I've shown you some examples. There's many biological results which I have not reported here. Then cell OT is able to reconstruct individual cell responses of perturbation, and this allows us then to do comparisons between before and after treatments, which allows us to do new analyses. Good. So this data will become open soon. So I think in the next few months, please check on this webpage. Okay, there's something wrong. So, and now some general conclusions. So if you look at both parts, so I think one point I'm trying to make is that it's really important to understand what the problem is, what you would like to predict, and what is actionably for a physician, let's say, or for a life scientist, what you would like to predict, right? So, and I really would like you to encourage to closely interact with the physician or the life scientist and deeply understand what the problem is and which predictions really hold value, so worth doing, right? So often there is, and when you start these collaborations, often there is a good solution which solves the problem reasonably well, right? And like our light GBM case, right? So it does it reasonably well. But then, since we have understood it well, it generates a lot of new research questions which we can look into. Why, how do we do with hospital transfer, or how can we improve the performance generally? So there's interesting methods questions which appear along the way, and these can lead to better solutions. So for me, this is often a cycle where we collaborate, get new ideas for new problems, and then we continue that cycle. So I think it's really worthwhile doing that. Then maybe one point, don't stop when you have a nice model, right? Demonstrate the impact, because only then really it has that impact in many cases. Often it is not sufficient to develop the method, throw it over the fence, and hope that somebody takes it, right? So one sometimes has to do it, like this clinical trial we are doing. Then I've seen many deep learning methods are reported, but they do not compare against the classical machine learning techniques. And it's, if you do them as a baseline, often they don't look very, very good. So do that. And biomedical foundation models are a promising direction, but I think they still need a lot of work. And I see this as an opportunity, of course, right, as a machine learner, to do that. But I think we have to convince that the simple methods really, I mean, can be outcompeted with these new methods. Well, and then maybe I showed you some examples of distribution problems, distribution shift problems in healthcare, but also in life sciences, from one patient to the other, one hospital to the other, or even from one experiment to the next, right? So, or one cell line to the next. And I think a more methodological research is needed here, how to deal with that and how to cope with it. It's not saying it's easy. Good. With that, I would like to conclude. This is my group, maybe a year old or so. There's funding sources, and I would like to thank you for attention and happy to take any questions. Thank you.
Thank you very much, Gunnar, for a wonderful talk. Okay, so we have time for some questions.
Very inspirational slides. I have 2 technical questions. So regarding the clustering and the latent representation, did you try some contrastive methods or some info NCE or some self-supervised learning methods? You use the triplet loss, right?
Yeah, so you talk about this part. Is just let's go there, right? So, so this one,
Yeah.
Right? Yeah, so we do some clustering first on these image features or the gene expression features, and then this allows us to do a triplet loss on top of that. Does it answer your question?
Yeah,
Maybe not.
I just said, did you try? Yeah, I see this triplet loss function is a bit somehow like successor of it is maybe some contrastive methods or some self-supervised learning. I just asked if you try that, but anyhow. Another maybe more interesting question is about distribution shift. I saw the slides about the visualization of more some prominent variables. You have some nice interpretation and visualization. My question is, did you find some effective solutions to address or to alleviate this distribution shift? That's a big topic, I think.
Yeah, I mean, we think about it, and we tried a few things, and I also tried to do something to include in this talk here. But I mean, okay, let's see. So this one here, right? So we tried a few basic things. So we checked, of course, whether the distribution of the variables, the distribution of the values for each of these variables is the same. And it looks reasonably similar, right? So we tried something like quantile matching, quantile transformations, and so on, and it all didn't help. So then we thought, okay, maybe we need some more complex method. We tried even cell OT to maybe transform the distribution from one domain into the other. It's complicated because there's a lot of...
Form, by the way.
Say again?
How did you transform?
I mean, essentially, you could say each data point here, you could, I mean, in the cell OT space would be a cell, right? So it would be a vector, and we would like to match distribution in the two hospitals. So that is one way to do it. But there's a lot of complications with discrete features, which are not handled very well by cell OT because it's non-differentiable. So, but that would be one approach which could work. There's other approaches like based on MMD, where you find weightings maybe of examples and then retrain the classifiers and so on. But so far, I haven't seen anything super convincing. So the best so far was identifying the variables which are responsible for the reduced performance.
Yeah, my research is about like images, and it has same problem, and it's actually a very important task in clinic, but actually technically there is not so many methods exploring this distribution shed or domain gap. So this is really I mean, for images, maybe a neural optimal transport potentially work to match the distributions. Maybe it's worth trying. So for this data, because there's many discrete features in there, it was hard to use. But yeah, but I would say it's an open problem. Maybe some augmentation could help to make it more robust. But yeah. Thanks so much.
So when you were talking about like the different methods that you use to evaluate the cancer patients, and that you can take a small subset of those and get the vast majority of the insight, and as a means of kind of a...
So you mean this where we yeah, right. So this one.
Yep. This
right.
one exactly.
Yeah.
So I kind of fear that this kind of figure might be misinterpreted by people looking at this and saying, oh, so the vast majority of the relevant information is in those few methods, whereas in fact, the vast majority of the interpretable information is in those methods, and that's very much affected by the fact that whole exons are something we've been doing for a couple of decades now. So we know that like if there's an EGFR mutation, we have targetable drugs for it, etc. Whereas single cell methods or spatial transcriptomics are very, very new, and we have very little actionable items there. So it's not like the data is necessarily less relevant. We just don't know how to act on it.
Totally agree. Totally agree. So I mean, obviously, if you have all this information, you can do much more with it in theory. So, but this is more the question: if I would do a clinical study tomorrow, right, and I would need to decide which technologies to take, then this could help me, because within the trial, I don't do new research to find out what the markers are necessarily, right?
Right.
I need to fix this in advance. But if I would like to do research, then of course I would like to have all the technologies.
My theory is that the person who wants to do research would look at this chart and say, oh, okay, so I don't need to do any single cell stuff or anything like that because it doesn't affect any clinical decision making, so there's no point in looking there.
Yeah, no, I agree. So I guess the interpretation needs to be taken care of.
Thank you, Gunnar. Let's maybe take the questions after this so that people can leave if they need to. Okay, so thank you very much for a wonderful talk.